This is Unit 6B, Part 1. I mentioned before to you that there are two parts to the immune system. There is the innate immune system and there is the adaptive immune system. And the innate immune system is non-specific. It's active right away. It doesn't need a period of time to undergo an activation process. And the other immune system or immune defense system is the adaptive immune system, which is very, very specific. The cells involved in this adaptive immune system are T lymphocytes and B lymphocytes, or also called T cells or B cells. And each of them are very specific and have specific antigen receptors on their surface. And they take some time to get activated and develop. Now, the innate immune system is nonspecific. It responds very quickly. It depends on which source you look at, but quite a few sources describe these as lines of defense, how we defend ourselves against entry of any foreign molecule, anything that's non-self. And that, of course, includes infectious microorganisms. So the very first line of defense, which is considered part of this innate or non-specific immune system, is the intact epithelial membranes that cover all of your surfaces, separating your body tissues from the rest of the the outside world. So of course on our outer surface we have skin, which is a stratified squamous epithelium. We also have stratified squamous epithelium on interior epithelial surfaces where there's friction. So in the throat, in the esophagus, in the vagina, in the end of the gastrointestinal tract, in the rectum, you have stratified squamous epithelium. And in other places you have mucosal epithelium on other inner surfaces that separate the outside world from your body tissues, like the rest of your GI tract, your genital urinary tract, your respiratory tract. All of that is typically covered with a mucosal membrane, and that acts as a barrier, a membrane barrier to protect you. It's an important block or barrier to prevent entry of microorganisms into your body tissues. The second line of defense of the innate immune system is made up of cells, white blood cells like macrophage monocytes or granulocytes, also called polymorphonuclear cells. Remember, there are three types of granulocytes. There's neutrophils, basophils, and eosinophils. All of those are part of that second line of defense. You also produce, mostly on the mucosal membranes, mucosal epithelial membranes, you produce antimicrobial proteins. These are very, very small proteins that have a direct antibacterial effect. There are also some proteins in secretions in the saliva, tears, that's called lysozyme. These also can directly kill microorganisms. Now, part of that second line of defense is the inflammatory reaction. It's a hallmark of that second line of defense. It's an extremely important mechanism. When inflammation is triggered, many different players, different types of cells, different biochemical compounds, molecules, come into play and act to try and destroy or kill whatever gets into you that's non-self. So, as I said, the first line of defense is the stratified squamous epithelium of the skin. mucosal epithelium or mucous membranes on inner surfaces, and also the secretions from that. The skin itself, the stratified squamous epithelium, as the epithelial cells of the skin form, they replace each other, and the dead cells and the new cells push up from beneath the older cells, and those older cells eventually die. It's a multi-layered barrier or layer of epithelium, and the topmost cells are flat and dead, and they contain a major protein called keratin. So the cells have died, but the keratin, which is an insoluble protein, is still there, and it forms quite a very strong barrier against entry of microorganisms. It's also resistant to weak acids and bases and any enzymes that bacteria could produce. Also prevents entry of toxins. The mucosal epithelium also provides a mechanical barrier, not actually quite as strong as keratin in the skin, the exterior skin, but it also, in addition, has secretions that contain these antimicrobial peptides. For example, if you think about it, if you had a cut on your skin, for example, and you kept it moist all the time and covered it and sprinkled a little food there, you would end up getting quite an infection. But when you get a cut in your mouth, which is warm and wet and full of food often enough, it heals very quickly, and that's because, and doesn't get infected, and that's because of those secretions that you have in your saliva and your mucus that you're producing on those mucosal membranes. Okay, the other aspect, of course, of the second line of defense are the cells, the phagocytic cells. The two main types of phagocytic cells in the body are the monocyte macrophages. Remember, when monocytes are in your bloodstream, they can leave the bloodstream and go into the tissues and then become macrophages. And the other type of phagocytic cell that you have are neutrophils. These are highly phagocytic. They can engulf foreign molecules, foreign particles, including microorganisms, and take them in and destroy them, kill them. They are also they form a very important main part of the inflammatory response. The inflammatory response is, in fact, characterized by 4 what are called cardinal signs. They include redness, heat, swelling, and pain. And they were originally described in Latin, and redness is rubor, heat is calor, swelling is tumor, and pain is dolor. And anyone who knows any Latin-based language would be able to figure out what these words mean, I imagine. So rubor, calor, tumor, and dolor are the 4 cardinal signs of the inflammatory response. Well, these phagocytic cells that you have, macrophage, monocytes, or neutrophils, as I said, can phagocytose. They will project out part of their cell membrane and cytoplasm around a foreign target molecule and take it in. So it gets taken into a vesicle, a phagocytic vesicle, properly called a phagosome. It will then fuse with another membrane-bound vacuole or vesicle that's present inside cells that are full of digestive hydrolytic enzymes. These are called lysosomes. So a phagosome that is filled with something foreign that was engulfed will fuse. Remember, these are membrane bound, and this is membrane bound here. Wait a minute, I'm sorry. The phagosome is membrane bound, and the lysosome also is membrane bound, and the two will fuse into one single structure called a phagolysosome. And following fusion, due to the action of the different enzymes, it will break apart and degrade whatever molecules make up this foreign body. And if it's a microorganism, it'll be able to kill it, because you get production of very toxic molecules that are called oxygen radicals. Anything that can't be broken down, that is still solid, will be ejected, because the phagolysosome will go on to fuse with the surface membrane and release all of this by a process called exocytosis. It'll release anything that wasn't that it didn't break down into soluble molecules. All of that, the phagocytic activity, the activity of the phagocytic cells, all of that forms part of that inflammatory process that I told you about. I wouldn't expect you to remember the name, but it was described by Celsus, who was a citizen of Rome, a Roman citizen living in between 25 BC stands for the before the Common Era. It's also called BC, or to 50 AD or CE, the Common Era. He described those four characteristic cardinal signs of the inflammatory response, as shown here. And then not too long after that, a fifth sign was described by Galen, who lived, well, it's about 80 years later. And again, you don't have to know the names or the year, but you should know the names of these four key processes of inflammation, and the fifth one, which is loss of function. So with all of these occurrences, these inflammatory responses, you get typically a loss of function. And everyone experiences inflammatory responses somewhere on their body at almost every time. You have a tiny little cut and it gets infected, or some dirt gets into it, even if it's not infected. If you have a foreign body in your hand, it will trigger this kind of inflammatory reaction. If you have damaged tissue, you have death of your own tissue, you'll get an inflammatory reaction associated with it quite often. The thing about the inflammatory response is it's very important, and it needs to be turned on quite quickly. It needs to be active immediately when needed. But the trouble is that it's so potent that it not only can damage and injure and kill microorganisms, but it can damage the tissue surrounding it. And therefore, normally it needs to be also quickly turned off. And there are a number of different mechanisms at the cellular and molecular level that will turn off the inflammatory response. Unfortunately, sometimes it has to continue because the microorganism that got into you is winning the balance and is persisting. So as it stays there, the inflammatory response intensifies. Here's an example of a pin being stuck into an epithelial. This is stratified squamous epithelium here. Let me get it a different color. All of this is stratified squamous epithelium from the lower layer here, where the epithelial cells are dividing and then pushing up in this direction till they form that layer of dead, dried keratin. So this is pierced through, and with it, it's brought some bacteria. These are bacteria here, and that triggers release of molecules that will attract white blood cells. This is this particular cell would be a neutrophil. I can tell by the shape of the nucleus. This cell here, I would say, is a macrophage. So cells will move out of the local capillaries and be able to squeeze between the endothelial cells that line the capillary and move in towards the area here where there's tissue injury and foreign molecules, including the bacteria. So it's attracted to it. It's this process of attraction of these cells is called, I think I'll get a better color than that. It's called chemotaxis. Not that easy to write with this. Chemotaxis, the movement of cells towards a concentration gradient, to the higher concentration of these molecules that are present here. Okay, chemotaxis. Now, I think I've described, oh yes, the one thing that I want to mention is that the process of squeezing out of the blood and into the tissues between these endothelial cells is called diapedesis. Okay, this is a flow chart which I haven't described everything here, but a lot of the processes I have described. I'll go through not all of it, but quite a bit of it. So with the initial tissue injury, you have release of some chemical or activation of some chemical compounds that are normally present, and they get released when you have some tissue injury. We don't have to go into the specifics of each of these necessarily, except for maybe histamine. That's an important compound that does get released. Now, what histamine does is it increases capillary permeability. So it increases the vascular permeability. There's an increased ability for fluids and cells to move out. The other thing is that it will trigger vasodilation of local arterioles. So you not only get increased capillary permeability, but an increase in blood flow in the area because of the vasodilation. These molecules, particularly complement molecules, can attract phagocytic inflammatory cells by the process of chemotaxis. Now, if you have vasodilation of arterioles, you will have more blood flow into an area. And you know that when you have an inflammatory process, you get redness, rubor, and you get heat, calor. It feels warmer to you. The reason is because there's more blood flow into the area, and blood is red, and also the blood is at a higher temperature always than the average body temperature. Your blood is just slightly warmer than the average body temperature of 37. It's about, say, 38, maybe a little higher. And so you will feel the fact that there's increased blood flow into the area, local hyperemia. You'll get heat and redness, two of the cardinal signs of inflammation. The other thing, of course, you have increased vascular permeability, so you will get swelling, tumor, swelling of the tissue. It's edema. It's increased flow of fluids out of the vasculature, and some of these molecules that are released will trigger perception of pain. And of course, in the end, you can get a loss of function. In this case, they're suggesting a temporary limit of a joint. I guess they consider this joint. But loss of function typically follows the inflammatory, accompanies just immediately after the inflammatory response. Okay, let's move on. So that was the innate immune response. The first line of defense was skin. Second was the phagocytic cells and some antibacterial molecules, and the inflammatory response, a key feature of the innate immune defense system. Now, the other main part of the immune response, or immune defense response, I think that's what your book calls it, but the immune response is the adaptive immune system. And that forms the third line of defense. And it is made up of cells that have very specific receptors for foreign molecules on their surface. T cells, or T lymphocytes, and B cells, or B lymphocytes. The T cells have specific foreign target antigen receptors. Remember, this is a foreign non-self molecule, so they have receptors for that. And the receptors are called the T cell receptor. Okay. Similarly, B cells have specific foreign target antigen receptors, and those are called antibody molecules, okay, abbreviated AB. So the T cell receptors, the TCR, and the antibody molecules, AB. These are not soluble antibody molecules. These are embedded in the surface of the cell membrane of the B cell. So they have the receptors all over the surface. Each different unique B cell or T cell that you produce from the primary lymphoid organs, remember these are formed in primary lymphoid organs. B cells are formed and mature and become immunocompetent cells in the bone marrow, and T cells mature and become immunocompetent cells in the thymus. Each one has a unique receptor all over its surface, one type of receptor on each B cell, and that receptor is specific for one particular target antigen. The next B cell that gets produced will have different receptors, and in their case, in the case of the B cells, the receptors are antibody molecules. Same goes for T cells. They'll have each unique T cell that you produce will have one type of T cell receptor all over its surface that's targeted to one foreign molecule. That foreign molecule is a target antigen or antigen molecule. It is foreign molecule non-self. In the case of B cells, it will activate the B cell that has the right receptor. When a B cell gets activated, it starts to produce and secrete its receptor. Remember, its receptor are antibody molecules. So now the molecules are produced in large quantities and secreted. They're soluble, so they get secreted out. If it activates a T cell, if the T cell has a receptor for the foreign molecule, they don't secrete their receptor, their T cell receptor, but they do get activated. The thing is that the adaptive immune system is very specific because of those specific antigen receptors, the T cell receptor on the T cells and the B cell receptors on the, or antibody molecules on the B cells. They work together with the innate immune system. There are interactions between the two that I'll describe a little bit later. Highly specific. You know, when you have a receptor, the idea of a receptor is it binds its target. It will bind its target molecule. So it's all about interacting and binding the target antigen for T cells or B cells. So here you see a diagram with a B cell, and well, in this case they've shown 4, but it's hundreds on the surface. These are antibody molecules. They're acting as receptors for foreign antigen. On the T cell, these are supposed to be T cell receptors, each of these, okay, all over the cell surface. Let's just go back a little bit. I'm not 100% sure why I put this one here, but let's just go back and review the idea of production and release of immunocompetent B cells and T cells. Remember, the B cells are produced in the bone marrow, in red bone marrow, and released, well, and then they're immature lymphocytes at first, and they stay in that bone marrow and they mature into immunocompetent cells, and then they leave. Okay, so B cells develop into immunocompetent cells in the red bone marrow, and then they leave. So the red bone marrow is the primary lymphoid organ, and then they go out and they go into the bloodstream and they'll seed into different secondary lymphoid organs like you know, the lymph nodes, spleen, the tonsils, Peyer's patches, places like that. The same time that immature T cells also get produced in the bone marrow, but then they migrate out and go into the thymus, and it's only in the thymus that they become immunocompetent, and then they leave. And just like the mature B cells, the immunocompetent B cells, they populate the blood and the lymph nodes, spleen, etc., other secondary lymphoid tissues. And they can move around. Both B cells and T cells can migrate. They're not stuck inside these secondary lymphoid organs. They can go back into the bloodstream and back to where they came from even, but they can move around the body. Okay. There's two important processes that I would like you to understand. One is the idea of self-tolerance. The body doesn't normally have any B cells or T cells that have receptors for self. B cells have receptors for foreign, non-self molecules. T cells have receptors for non-self, foreign target molecules. All right. The antibody on B cells is not directed to self, ever. The T cell receptor on the surface of T cells are not directed at self. If they were, then it would be an autoimmune response attacking self. We would have autoantibodies from B cells, or we would have the T cells attacking self. We don't. We are self-tolerant. Tolerance actually is developed during the development of the fetus. And if it breaks down, if we are not tolerant to self, then we develop an autoimmune response and we have an immune reaction to self. Now, the generation of T cells and B cells is of the receptor rather on B cells and T cells is a random process, and so every new T cell comes out with a unique kind of receptor on its surface, at least for the binding site, what it can bind to, and the same for B cells. But occasionally, actually probably more frequently, not that occasionally, sometimes a B cell or a T cell is produced which can react to self, can bind self, but they are destroyed before they get released. And that elimination of autoreactive or self-reactive cells that can react to self molecules, self-antigen reaction, that is called clonal deletion. So these cells are deleted. They are destroyed before they can get out of the primary lymphoid organs. And the process of destruction is called apoptosis or programmed cell death, which these two are synonymous, programmed cell death and apoptosis. And that's the end of Unit 6B, Part 1.